Thanks a lot for the nice introduction, and the organizers for giving me the opportunity to talk about our work. So I will talk about the distinct genetic architecture for syndromic and non-syndromic CHD. First of all, there is no relevant relation to any commercial organization at the moment. Let me just firstly go briefly over congenital heart disease. As most of you know, we have severe and less severe condition, atrial septal defects, VSD as the less severe ones, and then we have fallow and hypoplastic left heart, for example, as the most severe ones on congenital heart disease. Overall, it's the most common birth defect, and most of you are well aware of this. We have about 5 to 10 cases per 1,000 live births. A specifically interesting group are those ones who have life-threatening types of CHD. For example, those severe conditions which need catheterization or operation right after birth. And these are approximately 25% of the cases. And these are also the focus of my talk mainly, because they make up high infant mortality still, and they use up a lot of resources in the clinical setting. One really interesting or necessary distinction are those ones where you not only have a congenital heart defect, but where you have also extracardiac abnormalities. For example, developmental delays, or abnormalities of other organs. So these are called syndromic CHD, and it's about 20 to 30%. I think numbers are changing over time as we get more precise with the phenotyping. Numbers are changing, and this group will grow over time. Causes are different ones. Genetics, as we are in a genetic session here, and is the most common cause, but environmental factors and infection do play a large role. In mice, we have approximately 800 genes known to cause a congenital heart defect. And in humans, we have about 354 unknown, but there are more coming every day, and the literature is increasing steadily. The interesting thing is that the majority of those are the syndromic genes, although they are the minority of cases. As I said before, they are approximately 20 to 30% are syndromic. We have only a few genes for non-syndromic CHD, although these numbers are also increasing. What we have done over the last couple of years, we have set up a couple of international large-scale collaboration together with partners from all around the world. One was an exome study based basically on de novo mutation in the trio setting. So we had parents and the parents and the child. The parents were unaffected, and we're looking for de novo mutation, and we had cases from the non-syndromic side and the syndromic side. And as a follow-up of this, we did a large meta-analysis combining de novo mutation with copy number variants. As we have already seen in the previous session, copy number variants are important for CHD to find novel gene and associate them with congenital heart disease. In addition, we are now also working on a large-scale exome analysis with about roughly 5,000 at the moment, from the non-syndromic and syndromic size, and comparing them to 50,000 UK biobank control to really understand what are the driving forces and how CHD develops. So the identification is necessary really to have these genes genome-wide significance so that they are clearly associated also for the clinical purposes, and really also understand the difference in the genomic architecture and the underlying pathways. So these are the collaborating institutions. Let me just firstly start with our study from 2016, where we could show, as it's shown here, that we have an axis of de novo mutation in the syndromic and non-syndromic setting. So above are all genes here in the left side, and you have them specified by non-syndromic and syndromic. Those are the silent mutation, the so-called synonymous mutation. There's no enrichment for all for all genes, for the genes with developmental disorder or congenital heart disease genes. But what we saw at the time was that we had enrichment for non-syndromic and syndromic protein truncating genes. And this was really high for the developmental disorder side, where we had in the syndromic category, we had a large excess of protein truncating mutation. And this was even more pronounced in the syndromic side on the known CHD genes. Similarly, we saw a similar picture for missense mutation, which was also present in the non-syndromic side, but was most pronounced in the syndromic setting. When we look at inherited mutation, which was possible at the time because we had parents and unaffected parents and the affected child, we didn't find any enrichment in the silent mutation, which is a good proof of principle that it worked. But we did find for the protein truncating mutation in the non-syndromic setting here on the left side, as an enrichment for CHD genes in all genes, pointing to an observation that possibly healthy parents do pass on mutations from both sides, which kind of makes the congenital heart defect apparent in the child. So pointing again to a more oligogenic nature in this non-syndromic gene setting. So at the time, we were able to classify a lot of candidate genes to CHD, protein truncating mutation. We had about 25 candidate genes at the time. Now they are proven genes to cause congenital heart disease. Several of them had already been known, 8, but there were a couple of novel genes which are shown here as an overview. What is interesting is that most of these pathway or the genes involved in certain pathways were known pathways, like the erythropathy pathway, PTPN11, SOS1. Those are the classic one. But we also had a lot of overview with the Notch1 pathway, Notch1 signaling EP300 on the histone modification side, which has been also shown by other studies, and including ours. So from that, we went on to do a copy number de novo mutation meta-analysis. So we do know that de novo mutation show a strong enrichment for the previously shown pathways. So then the question was, do we see in a case control setting, we collected data from about 4,000 cases with copy number variants on CHD. And we had about 12,000 control from the Wellcome Trust Case Control Consortium. And we're interested, do we know what is already known in the de novo setting? So do we find enrichment in those copy number variant regions or genes, because we did it on a gene-wise basis for the known genes, and we did. So the syndromic genes, as shown on the left here, show a really high enrichment. The non-syndromic didn't show anything. And then you had monoallelic gene, which is not surprising, biallelic showed a small enrichment, developmental disorder, and the known haploinsufficient gene. So this approach made us certain that we are on the right track. In addition, we looked at genome AD, looking at those genes which show only a really rare occurrence of severe mutation. This is called observed over expected ratio. And we found the strongest enrichment on the right here for those genes where you have only rarely find protein truncating mutation in these genes. So this made us certain that we are able to combine the de novo setting with the copy number case control setting. And this, we used the Fisher combined method, and we could use the expected over observed ratio as a sort of hypothesis weighting to increase your likelihood that these genes are actually causal. So this study was further we further used a prioritization approach from human bulk RNA-seq data. So there is a really nice study where where they have taken human fetuses from different stages of development, different stages of heart development, from early development to maturation and infant. So the question was, are our genes, which we now find in this combined setting, also enriched in early development or in maturation and infant cardiac transcript, which indeed was the case? We found strongest enrichment in development, which points that these genes are clearly implicated in development, and this allowed us to further characterize or prioritize these genes. So with that, we were able to identify 21 haploinsufficient genes, either which have been significant on the de novo side or both with the copy number variant side. And a lot of them had already been known and had already been associated with other studies, but 7 were unknown or had only limited association, meaning that they have only been described in single patients, single families so far. What was more interesting, this study also allowed us to look into the distinction between LVOTO and conotruncal, if specific genes had sort of an enrichment in the LVOTO setting, meaning aortic stenosis, hypoplastic left heart, and coarctation, versus conotruncal defects like, for example, tetralogy Fallot. And that was indeed the case. So we had KMT2D and KMT2A as well, TAP2. They were enriched for LVOTO, whereas NSD1 was enriched for conotruncal, which is interesting. And that's something what we are currently following up to see if the surrounded networks also show an enrichment for these LVOTO versus conotruncal. What we found is that we had certain gene clusters, 10, which were significantly enriched. One of the most interesting one was the Notch1 cluster. So Notch1 has definitely been shown to be a really important gene in congenital heart disease. And it has been shown on the LVOTO as well as on the RVOTO setting. So here you can see that the controls versus cases, you had a strong enrichment for Notch1 mutation and copy number variant in Notch1 for these cases, and you have pulmonary valve development and pulmonary valve morphogenesis as sort of highly enriched here. So with that in mind, I want to move on to the next big study we used. This was the exome study with about 4,800 cases, and we did a case control setting, focusing here, you can see an overview, focusing really pretty much on only Caucasian cases to make sure that we have no spurious results in regards to population biases. So this study, again, we have enrichment for the known developmental disorder genes. We have here on top the loss of function high confidence genes, protein truncating mutation. MPC2 is a score which allows you to prioritize missense mutation. They behave similar to loss of function, like missense badness mutation. And CAT score is another score to also prioritize missense mutation. Synonymous, we had no enrichment. Similar picture for haploinsufficient gene, also strong enrichment for the high confidence loss of function. Missense variants also, but not as pronounced in the previous one. Nothing in the autosomal recessive setting. In the non-syndromic genes, we found an enrichment for the MPC2, for the missense badness. Pointing again to something we observed also previously and also other studies by Wendy Shang recently, that there is evidence that some of these missense mutations, maybe as combined as oligogenetic, make up a large chunk of the or explanation for the non-syndromic congenital heart disease. For the syndromic genes, also MPC2, CHD showed a really strong effect, and you have to keep in mind that this study is mainly syndromic cases, so there is a really strong aspect in this. And then for the monoallelic genes, where you also have the syndromic side being really strong, and the biology, where you had almost no enrichment on all of them. So what was the most obvious and most easy scenario we looked at was the protein truncating side in the case control setting with the allele affected 0.1%. And we do find in the syndromic on the left side as well on the non-syndromic on the right side, we do find the known CHD genes like KMT2A, Ankylin D11, NSD1, which we have previously also discussed. What is more interesting, on the non-syndromic side, Notch1 is by far the most significant gene in non-syndromic. And it's probably explaining in our cohort, and there are several studies showing this again, 0.5% of the CHD cases. So that brings me actually to the patients with Notch1 in our cohort. And it is known that Notch1 causes Adams-Oliver syndrome. That's a syndrome with aplasia cutis congenita, terminal transverse limb defects, and 23 of the cases have a major congenital cardiac abnormality. So we were interested, how does the variant spectrum in our cohort and also in published cohort look like? This is our cohort basically. And do we find sort of enrichment for specific phenotypes within notch one? So are variants at the end or the beginning more likely to cause LVOTO versus RVOTO, for example? So we had about 27 cases versus three controls. It's a bit different on the variant level because we had also multinucleotide variant, which makes it more interesting, more defined. So some complex variant where you had sort of repetitive indels in this cohort. One interesting thing was then to look at the reports if we find any overlap with Adams-Oliver. And to our surprise, this was really limited. So when you look at the typical syndromic features of Adams-Oliver growth defects or limb or malformation or any others, we could only find a small, and brain abnormalities, we could only find a small proportion of the 27 showing any or even only minor signs. What was more striking was this part here. We found that many of the LVOTO cases actually had an enrichment in the intracellular domain. So this might be of interest in the future, and it's pretty really important to do detailed genotype-phenotype follow-up to really define this more clearly, because we also know some families where we have RVOT and LVOTO. So what is more interesting is that the majority of cases are RVOTO cases. So you have hypoplastic right heart, double outlet right ventricle in 60% of the cases. So this is clearly not an LVOTO gene. It is mostly an RVOTO gene, which many of them having TOF or TOF-like phenotypes. So more recently in our in silico analysis, we could also show that those mutations in disulfide binding site stores really strong enrichment for RVOTO, which is not shown here. So with that in mind, and to keep it short, I would like to briefly summarize what we have done and what I hope I could show what is significant here, that we have shown that we had about 25 genes which were enriched for cardiac congenital heart disease cases, that we have a difference in de novo mutation syndromic versus inherited mutation in non-syndromic cases, and that the CNV meta-analysis allowed us to reclassify novel genes actually to congenital heart disease. 7 of them have not or had only limited association previously. And also, which is more interesting for follow-up, is those enrichment on LVOTO versus chronotruncal for specific genes. On the case control side, I think there's much more to learn, and I think we are getting there. And NOTCH1 is really the most commonly mutated gene in CHD, and it's really necessary to have clear genotype-phenotype follow-up studies on NOTCH1. Really to see where these different variants localize and how they behave. So this is what is underway, and with that, I would like to thank also my collaborators and the 3 main people who drove that work: Enrique Audin, Anna Wilson, and Gregor Dombrowski. From David Brooks' lab is Anna Wilson, and then of course Matthew Halls is one of my main collaborators, and then the other people from the congenital heart or German heart registry, where we got a lot of samples from. So with that, I'm happy to take questions.
Thank you very much, Mark, also for staying in time. And currently I see one question in the chat. It's a long question, so I'm going to read it. It's a question from Felix Boschan. Is there a correlation between disease severity and the likelihood of detecting a causative variant in the non-syndromic CHDs? Does this play a role with regard to the time of diagnostics, e.g. prenatal versus postnatal diagnosis?
That's a really good question, I have to say. So we haven't checked that. So one thing we need to do, and that would be possible within the German Heart Registry and the collaborating institution, to see when they are diagnosed. We have to keep a bit in mind that we are also a bit biased and related to the phenotypes we've chosen. We have really chosen the severe onset phenotypes, those prevalent at the below the age of 1, and mostly with the necessity to have catheterization or operation. So there is this bias, but I think that is something we have to do in detail. We're following this up in another consortium more on the follow side, actually, together with colleagues in Toronto, Sima Mittal, and Amsterdam. But I think there is something, but we haven't done this. People try this a lot in the setting of prenatal testing, but there was nothing significant so far. So, but I guess there is something.
Thank you very much. I do not see another question, but I might have one. I really thought that's interesting, this Adams-Oliver phenotype, or you saw many variants that are actually then explaining the cardiovascular phenotype. So is this something you will follow up on, or do we really need those large cohorts to find the correlations?
Yes, we do follow up on this. So what we have done actually is we did methylation profile on these cases, and which actually interestingly allowed us to classify some of the disease entity a bit better. So that's follow-up work. We also cloned like all of these variants. Mm-hmm. Some of them are actually overlapping with known Adams-Oliver variants, which is interesting. Why do you have an Adams-Oliver phenotype in one case, and why do you have only a mild congenital, like not mild, severe congenital heart defect without any extra cardiac anomalies? So I think there we have doing assays like luciferase assay, testing these variants in the cell setting. But I think we probably need like those large cohorts to really be precise in genotype phenotype correlation. And I would guess there is also the epistasis, like other genes, playing a major role in this. So if the JAK1 is also sort of not functioning in the right like in the right tissue at the right time, that might be an explanation why you have more Adams-Oliver related to only having congenital heart disease.